Genes are linked to proteins, including enzymes. The one gene, one enzyme hypothesis. One important reason for the persistence of the modern synthesis is the success of the one gene, one enzyme hypothesis proposed by Beadle and Tatum in 1941. In 1941, Beadle and Tatum published their landmark experimental paper illustrating their success of deducing biochemical pathways in eukaryotic organisms by analysis of Mendelian mutations. This was made possible by clever experiments that relied on the fungus Neurospora as a model system for controlled experiments. This represented a considerable advance over analysis of biochemical pathways in prokaryotes. In the cautious style appropriate for reporting the results of pioneering studies, Beadle and Tatum wrote, "The preliminary results outlined above may offer considerable promise as a method of learning more about how genes regulate development and function." This was followed with their important insight. For example, it should be possible by finding a number of mutants unable to carry out a particular step in a given synthesis to determine whether only one gene is ordinarily concerned with the immediate regulation of a given specific chemical reaction. These preliminary results were borne out by subsequent experiments. From one gene, one enzyme to one gene, one protein chain. Following the discovery that DNA was the genetic material, subsequent discoveries in molecular genetics in the 1950s and 1960s led to a concrete understanding of the structure of DNA, the genetic code, and protein synthesis. Genes were shown to contain sequences of DNA that determine the sequence of amino acids in a variety of protein chains, not just those found in enzymes. As discussed in Chapter 4, each protein chain is a linear sequence of covalently bonded amino acids, where each position in the sequence is one member of a set of 20 possible amino acids. Each codon is one of 64 possible nucleotide base triplets in the DNA strand, 4 by 4 by 4 possible nucleotides per triplet. With the new understanding of molecular genetics, the one gene, one enzyme hypothesis, a powerful tool for deducing biochemical pathways in eukaryotes with genetic experiments, was expanded to the broader one gene, one protein chain paradigm that is now well established. The significance of protein synthesis and protein chains as timeless innovations was discussed in Chapter 4. Protein chains represent fundamental units of structure and function. They are involved in virtually every molecular interaction that governs the inner workings of cells.